we actually stopped at pop K. Can you see the screen?
It's better you go ahead because when you share the video, it will be clear. Just go ahead.
Okay. So we actually stopped at
We
this.
cannot see the screen.
We actually stopped at Atenolol, where I've said you can perform different kinds of search. So when you typed Atenolol, the first result that comes out is for the best match. So that is, so you can pick this best match. You can click on this best match if you are looking for only informations about atenolol. But then if you want similarity informations, you can click on this similar structure search. This is similar structure search. So once you click on similar structure search, what you're going to see is identity, similarity, substructure, superstructure, 3D similarity. So under identity, we're having 1 under identity, which means there is only one compound that is atenolol. So if you click on identity, you see only one result, that is the best match. When you click on similarity, what you are going to see as a result of similarity are structures that are similar with atenolol, compounds that has a similar component, similar structural features with atenolol. That is what you see under similarity search. And we have about 564 compounds that has similar structure with atenolol. Then substructures, we have 410 substructures. These substructures has to do with structures that have the same scaffold with atenolol. The difference is just the functional. The difference is just their substituent. That is the only difference. But they maintain the same scaffold with atenolol. So all these compounds are compounds that are derivatives of atenolol, which I've earlier explained. I just did this to show you what I was talking about. Then superstructures. I told you that superstructures are it shows the fragment of the compound you just typed in. So all these things are the bits, the smaller bits of the compounds that make up atenolol. We don't have interest on superstructures. It depends on what you want to do with it. So let's come back to where we are. We stopped at molecular modeling. So this was where we are. We just concluded database management. Now in molecular modeling, we can use chemoinformatics to predict molecular geometry. Please, if your mic is on, just help me unmute it so that we can get it clear. I don't know whose mic is on. So we can use another key area of chemoinformatics is the molecular modeling, where we can use it to predict molecular geometry and energy using force fields or quantum mechanics. We actually talk about that when we are dealing on energy minimization. When we are dealing on energy minimizations, we are using MM2, which is a molecular mechanics. So, but this is a bit more above our script for now. We can also do what we call docking. We can do docking online using chemoinformatics servers like SwissDoc. We can use SwissDoc for docking. We can use chemoinformatics tools for homology modeling. This homology modeling is when you are working with a disease target, and that disease target does not have a known protein available online, we can use homology modeling to build that protein. I will explain in details when we get to docking. Another key area of chemoinformatics is the QSAR and QSPR modeling. We can use chemoinformatics to build QSAR. QSAR simply means quantitative structure activity relationship, while QSPR means quantitative structure property relationship. Now what I mean is that if we have structures that look alike, it simply means that these structures that look alike can have the same or similar properties. That is the meaning of structure activity relationship. So if I have 50 imidazole derivatives, and these 50 imidazole derivatives can target ulcer. Now if I want to build a new derivative of imidazole, it simply means that since these 50 derivatives of imidazole can target ulcer, this my new molecule can also target ulcer based on the structure activity. That structures that look like imidazole, the activity is that they target ulcer. Now that I'm building a new structure of imidazole, it is also likely to target ulcer. So we can do that using chemoinformatics. We can do that model, we can build that model with our chemoinformatics. All what we just need is to add small statistics, small statistical model. We can use machine learning, we can use a deep learning, but for the course of this training we shall be using multiple linear regression, MLR. We shall be using multiple linear regression in Excel to build our model that can predict QSAR and QSPR. Now it's often used in drug discovery and toxicity profile prediction. The same thing also, if I have 50 derivatives of imidazole and these 50 derivatives has been found to have liver toxicity. Now that I'm building a new derivatives of imidazole from structure activity relationship, we can easily now predict if this my new structure can also affect the liver without me going to the lab to conduct liver function tests using animal model. So we can use your chemoinformatics we can apply it in drug discovery and in a toxicity profile prediction. Another area of chemoinformatics is called virtual screening. So these are the key areas of chemoinformatics. We have the virtual screening. In virtual screening, we are trying to screen large libraries of structures against a particular target. So here we screen large compound libraries to identify potential leads. So let's say for instance I went to drug bank and I downloaded 1 million compounds, and I want to do a work on hypertension. So I can use my chemoinformatics tools to narrow down these 1 million compounds. I can screen these 1 million compounds at a time on the hypertensive targets. So at the end, I can be able to obtain the lead. Although we move from library to hit, from hit to lead. These are terms in docking. So I can be able to get one compound out of these 1 million compounds that are best and effective for my hypertension. We can do that using chemoinformatics to do virtual screening. Then it will include ligand base because it's based on our ligand. In docking, there's also called ligand based docking and structure based docking. We have a LBDD, ligand based molecular docking, and ligand based drug design, and structure-based drug design. So, but when we are using chemoinformatics in virtual screening, it means we don't have business with our targets. Our business will be on our ligands. Our business will be on our ligands. So wherever we use our ligand, using known active molecules and structure-based approaches. So it can actually combine both your ligand-based drug design and structure-based drug design. But by the time we begin to look at docking, I always advise students to combine both ligand-based and structure-based drug design. Because ligand-based, we focus on the ligands, on the activities of your structures, while the structure-based will look at the ability of the structure to actually attack a particular disease target. So, but that will be when we go to molecular docking. Now, applications of chemoinformatics. We can apply chemoinformatics in drug discovery. In drug discovery, we can use it to identify hit and lead. Now, let me explain the meaning of hit and lead. If I have a library of 1 million compounds and I want to see these 1 million compounds, which of them can be used to perform my degradation. Maybe I want to degrade a particular or maybe say I want to design a work that I have 1 million compound and I want to use a given bacteria to degrade this 1 million pollutant. Now, if I'm using E. coli, for instance, to degrade the 1 million pollutant, that 1 million pollutant is a library. When I apply E. coli on this 1 million pollutant, the E. coli might have high result for about 10 of that pollutant. Now that 10 pollutant is called heat. It's called heat. So from that heat, I can then optimize it to get my lead. So my lead becomes the best pollutant that can be degraded by my E. coli. That is my lead molecule. So we have from library to heat, from heat to lead. And before you get a lead, you must perform what we call lead optimization. So we can apply chemoinformatics in drug discovery. I may have 100 compounds from a leaf. Probably there is this information that there is this leaf that can cure cancer. Soursop. There was a time I got to be informed that soursop can cure cancer. Now let's say for instance that this soursop has 100 compounds in soursop, and we're able to see, okay, these 100 compounds, which of them can be more effective on the cancer target. We may reduce that 100 compounds into 10 or 5. These 5 happens to be the 5 that are more effective. Those 5 is called heat. It's called heat. Now from that heat, I want to get a lead compound. So I will optimize all my heat to see among these 5 which one is more effective. The one that is more effective among the 5 or among the 10 becomes my lead compound. So we can use chemoinformatics in drug discovery to identify heat and lead compounds, which are all go in details as time goes on. These things might be sounding strange, but just allow the process to go. You will get used to it. We can use it in SAR studies. I've already explained about these SAR studies. SAR means structure activity relationship, whereby we have a knowledge that similar structures can do similar properties. Similar structures can exhibit similar properties. So we can do that in using SAR studies with our chemoinformatics. We can do ADMET prediction, very important. Do you know that in drug discovery, it is not enough that a compound can cure a disease? It is very pertinent and most important for you to look at the pharmacokinetics of that compound. how will this compound be absorbed by the body? How does the body react with the compound? That is pharmacokinetics. How the drugs react with the body is the pharmacodynamics. These two keywords forms the principles of pharmacology. So having gotten a drug, a hit or a lead that can be used on a given target, we can then use our chemoinformatics tools to get the absorption parameters, to get the distribution parameters, the metabolism parameters, the excretion parameters, and also the toxicity. Around March, sometime by March, I presented a paper on a conference on parasitology and public health, Southeast region. So they had a conference at IMSU, at Imo State University. So on that conference, I presented a paper where I formulated a new insecticide using organosulfur compounds that has sulfur and garlic, like ajoin. So when we did that work, we used 24 target sites for insects, 24 target sites, and our compounds happens to have the ability to kill these insects even at the larval stage. But we had a problem. We identify a hit, which is ajoin, but this ajoin had high level of skin sensitization and eye irritation. That means that we cannot proceed with that studies, because I've used my chemoinformatics to study the toxicity of these compounds. So whereby I don't need to be visiting the lab anymore, the information has been deposited or retrieved by algorithm online. So these are what we can use chemoinformatics to do in drug discovery. I've already explained about this one, which is toxicity modeling. We can use it to model our toxic profiles of a molecule. Another aspect which I look at is data sources. How do we get, I've been talking about database. How do we get informations? There are different kinds of database. So our major aim is to start picking one after the other. But meanwhile, we have this database classified. We have phytochemical database. These database are actually used for information that are related to plants. We have phytochemical database. We have synthetic or chemical database. Like PubChem is purely a chemical database. It's not a phytochemical database. I was telling you that Nigeria does not have a phytochemical database. We are blessed with a lot of indigenous medicinal plants, but we don't have a database that has curated information about these medicinal plants and their therapeutic uses. We don't have a database in Nigeria that can tell us each of our medicinal plants and their bioactive compounds. But in Indian, we have IMPAT. IMPAT, IMPPAT, is an Indian database of their medicinal plants, which once you just type in any plant, you will get all the information about that plant, including the bioactive compounds present in the plant, which has been corrected through GCMS results. So we have different classes of database in chemoinformatics. We have database for phytochemicals. We have chemical database. We have database for disease prediction. Now, if I have a compound and I want to know what kind of disease this compound can treat, we have Swiss target prediction. Swiss target prediction is another database that can predict disease targets. I think at this point, let me be showing you. As I'm calling them, I'll be showing you, because we shall be picking these data one after the other. I was talking about impact. This is impact database. At this point, I'll be releasing some of the database we shall be using to work as the time progresses. How do I get these data? I just explained that one of them is from phytochemical database, and one of the phytochemical database which we shall be looking at is impact. Impact is owned by India. This is impact. Impact is a phytochemical database. It has about 17,967 phytochemicals. Another phytochemical database that we shall look at is Dr. Dukes. Dr. Dukes is a database owned by American. Dr. Dukes is a Chemoinformatics database owned by American. It is also under phytochemical, because I'm dealing with phytochemicals for now. It's also under phytochemicals, so you can use it to get relevant information about plants, about a plant. That is Dr. Dukes. Hope, can you all hear me? Are you with me? Hello, are you following? Hello? Oh. I want to know if you are following up. Okay. Let me just continue. So another phytochemical database is Dr. Dukes. Dr. Dukes is a phytochemical database. You can use it to source for information about plant species. Another one too is MPAS. MPAS.
Yes, sir. Yes, sir.
Okay. Another one is MPAS. MPAS is also another phytochemical database. These databases are owned by different organizations. Another one which I looked at is PhytoHub. PhytoHub is another phytochemical database. PhytoHub. I have a lot of these databases at my disposal, which we shall be looking at them one after the other. PhytoHub is a free source that provides information about polyphenols, terpenols, alkaloids, and other plant secondary metabolites in foods and in humans and animals. So outside phytochemical database, we have chemical database like PubChem. The PubChem we have opened earlier is mainly for chemical information, although we can still use it for phytochemical database, but PubChem is not ideal for phytochemical database. It's mostly ideal for chemical database. And let me say this, PubChem is a universal database that any structure, any new chemical species that is discovered is always deposited in PubChem. So if you have a structure and you search it in PubChem and you did not see information about that structure in PubChem, you can search in ChemBL. ChemBL is another chemical database. We shall be looking at each of them one after the other as time progresses. It's a very vast area. ChemBL is another database. is a manually created database of bioactive molecule. Now, ChemBL is not really for plant, but ChemBL is for chemical database. Now, ChemBL also can be likened for biological database. We can also use ChemBL to get biological information of our chemical species. So if I want to know that a compound is novel, if I type that compound in PubChem and there was no result, I go to ChemBL, there is no result. Just bear it in mind that you have gotten a new compound that has not been in existence, and that makes it novel. So ChemBL is another database for chemical database. We have zinc. Zinc is another database. There are so many of them, a lot of this database available to us. Zinc is a free database of commercially available compounds for virtual screening. Blood bank is another chemical database. Blood bank has all FDA approved drugs and their chemical information. Blood bank is a comprehensive and reliable source of pharmaceutical data for research, discovery, and software integration, which are all picked them one after the other. So we have another database which we are going to look into, that is SwissAdMe. Okay, we have database for disease prediction. SwissAdMe is, we have Swiss target prediction. Swiss target prediction is a database classified under disease target prediction. So I've classified them. We have a phytochemical database, we have a chemical database, and now we are talking about disease prediction database. So we can use Swiss target prediction. It works very well for disease prediction. So if I have a novel compound, I can just generate the smart code of that compound, come to Swiss target prediction. It will predict the possible disease target of that your compound. So that is for disease target. Now we can get information on the pharmacokinetics profile. So we have some database that are for pharmacokinetic profile, drug profiles. One of those database is SwissAdMe. SwissAdMe is another database for drug properties. Drug properties, you can use SwissAdMe, ADME, for such kind of disease drug informations. If you want to know if a compound can be drug-like, the pharmacokinetic properties, you can get that from SwissAdMe. So I'm just opening you up to different kinds of this database. As we meet on Thursday, we'll begin to pick one of them and study in details and see what we can get from this database. We have another one, AdMedLab. AdMedLab 3. AdMedLab is another database for pharmacokinetics prediction. It can predict the pharmacokinetics of your structure. There are thousands of Chemoinformatics database, which we don't know if we can exhaust all of them. But we can pick, we'll just pick the most relevant ones that we need for this study. Meanwhile, I want to show you something. Click I want to show you a comprehensive list of all the chemoinformatics database. Then you will see what chemistry has done for us. For those of you who want to explore more in chemoinformatics or in computational chemistry. Now, are you seeing my screen? Hello, can you see my screen? I want to explain something here. Can you see my screen? Hello? Hello? Oh. Okay. This is actually a chemical database. These are some of the databases used in chemoinformatics. There are a lot of them, tens of thousands of databases, which we may not cover all of them, but we'll just pick out the ones of interest.
Yes, we can.
So we'll just pick up the ones of interest. There are thousands of,
You can see it though. You're showing 2D drawing.
yeah, there are thousands of database
We
which
can see you. Me, I
which
can see.
you may not exhaust them. But what I'll just do for you is I will pick out some of them.
The screen is blank. I'm just seeing a blank screen.
I can see your screen.
I'll pick out some of
I'm
them.
seeing a blank screen.
The blank screen
I can't.
should be
Hello?
network. But let me reshare my screen. Let me reshare my screen so that others can see it properly. Okay, I'm coming. I want to reshare my screen so you'll be able to see it properly. So can you see my screen now? Can you see the screen? I think all of you now can see the screen. Okay. I think today's class has been well.
You can see it.
Okay.
Yes, you can see it. You can see the screen now.
Okay. So I was trying to show you some of the databases.
Yes,
Yeah,
we can
we can
see it.
see
You
it.
can see the screen.
Okay. I was trying to
You
show
can
you
see it.
some of the databases that we
Yes, so
exploit in chemoinformatics. I have shown you some chemical database. We have zinc, we have zinc 15, we have ChemBL, we have Spider, we have Cococo, we have Broadband, we have PopChem, we have TCM, we have Scribble Ido, we have Mucol database, we have Wombat, approved drugs, Spider Mobile, eDrug 3D, Glass, we have ChemDB, we have Express, we have ZincLeak, we have Express, we have Mod, KKB, Dudu, GPCR, so many of the databases available that has been created by scientists like you and I. So Visual Library Repository. So these are just a picture of most of the databases available within our disposal. So nevertheless, we have been able to look at some of them. We have classified them depending on what you want to work for, depending on what information do you want to get. We have seen ChemBL, we have seen Zinc database, Broadband, BindingDB, SwissAdMail, TOSNet. These are different databases. But then, what are the challenges in chemoinformatics? We will not just look at the glory of chemoinformatics and decide not to pay attention to the challenges. So we still have challenges in chemoinformatics. We still have some challenges in chemoinformatics. Challenges is quality and standardization of data. Most times, because we are collecting data from published work, because if we want to build our database, we usually take information from published works. But how sure are you that those works that we have published are authentic? Because what some of us does is that once it's time for appraisal and anywhere we see Jonah, we'll put our name. Some persons will just go online and cook up some work and publish. And those journal publishers will also publish it for you. And by the time we want to build our database, we'll start downloading the rubbish that you published, and we'll see that the work is not authentic. That is why if we are building a database, we have to look out for reputable journal sites. We use those reputable journal sites to build our database. So we don't have quality data. That is a very big challenge in chemoinformatics. Another challenge we have is handling of large data sets, big data. Most times, since we are not operating on cloud, because currently we are now trying to integrate AI.
Harry, please be time conscious, so they have time does.
How many minutes left?
Just 13 minutes.
13 minutes. Okay. We are almost coming to the end. Another challenge we have is handling of large data sets. We cannot be able to build a database that will have millions of data, millions of informations on our data. So that becomes a very big challenge to us in chemoinformatics, because in chemoinformatics we expect to work with large data sets. We're supposed to work with large data sets, but so handling of this large data set to be a challenge. Another challenge is accurate 3D conformer generation. Most times the structures we retrieve from some of these databases are not in 3D. Like PopChem, sometimes you see a structure in 3D, you also see a structure in 2D. So what you then need to do is, when you download a structure in 2D, we have to use our Open Babel to convert those 2D structures into 3D before we can work with them. So these are challenges in chemoinformatics. Another challenge is the integration with genomics and system biology, that is multi-omics. This area is much more common in bioinformatics. So most times we lack that integration between bioinformatics and chemoinformatics. Though Swiss ADMEL it comprises both bioinformatics and chemoinformatics, but we lack enough data sets that can incorporate informations on chemical species and also informations on biological data. So we lack such kind of data on genomics, data that provides genetic informations about our compounds without going to bioinformatics. Another challenge we have is interpretation of black box models for machine learning. It's just like everything I'm saying now is coming from my brain, but we don't know if what is being processed in the brain is right or wrong. The only thing we are seeing is the output. So it's a challenge. When you input a structure in chemoinformatics, you only see the outcome. You don't know the process that led to the outcome. That processing part is the black box. So the processing part is the black box. So we don't know what happens within the other part. It's a very big challenge, but I think this challenge has been solved. Some of them have been solved through the future of chemoinformatics. We have a future. Yeah, we have a future. Chemoinformatics has a future. And what is that future? We are aiming at integrating AI and deep learning for better prediction and design. I promise that in the second phase, we have another package called machine learning. In the second phase of our training that should start by February, we will not cluster all the programs the way we did like this. We will split them. So if you want to learn all, that is not your choice. We will have drug design, we will have machine learning and programming, we will have different areas, environmental, computational chemistry. So depending on the area you now want to apply your computational chemistry. So our future is integrating AI and deep learning, whereby AI will do the work of our programming and provide useful results. The future is cloud computing. In this case, we can now handle big data. Remember, we have the challenge of large data. We can do our computing on cloud, where we don't need your system storage. Some of you, very soon now, your system will be giving you red bar, telling you is storage full. But in quantum computing or cloud computing, there is nothing like storage problem. That is the future where we are going to. Expansion into green chemistry, personalized medicine, and synthetic biology. So these are the future of chemoinformatics. I want to appreciate you for being around till this moment. You have stayed, and it has not been so easy. You have really stayed, and you have proven your seriousness. The part we are in is a very serious part. Just like I told you, we shall be building a database. So we'll build a team. We shall build a team. I've already told the online, and I've told the physical students we're going to build a database, and we'll launch that database. Yes, we're going to launch the database, and so far I want to appreciate you. I want to appreciate you for being around. Do you have any question? Do you have any question to ask? Yes.
Yeah, good evening once again. I didn't join on time, but so far so good. I I think I got something from what you said. My question is very simple. You know, this database, you said something earlier which got my attention, and you just repeated it again. And what I want to ask is, how do we build this database? Is there any any computational development that is needed to achieve it? That is number one question I want to ask. Then another one I want to ask is in the quest of checking if a compound is a novel compound, you said if you search for that compound in so many databases that is not there, automatically the compound is a novel compound. So let's assume now that I worked on the existing compound and I tried to modify it, and at the end of the day, it happened that the exact uh, um, derivative of that compound has not been produced elsewhere. Meanwhile, it's not in any database. So how do I verify? How do I check to verify that it's not in any database, that the compound is actually another compound? Thank you.
Okay. Let me come from behind. Once you did a derivative of a compound, it means that you actually optimizing that compound to see better structures of that compound that can be effective. So if you want to test if that your derivatives is actually novel, you can take you generate the chemical identity of that your structure, like the SMI code. Some of this database works with SMI code, like the suicide mail. So generate the chemical identities of this SMI code of your compound. You can assemble up to 10 Chemoinformatics database, input the structures, either the name of the SMI code on these databases. They will tell you no result. Once you see no result, no result, no result, no result, no result, it simply means that it implies two things. It's novel. Yes, it's novel, it's new. Because where we are supposed to see the result of these compounds is not there. So it either means nobody has worked on it, or people have worked on it but has not deposited it. So it either means nobody has worked on it, or people are currently working on it at the back and has not deposited it. But once it has not been deposited on any of these database, it is novel. Like the compound I shared with you while we are doing synthesis and reaction scheme, the one I will use for reaction scheme modeling, is novel because I've tested it in about 20 different chemoinformatics and the result has not been found there. And the compound is not of a plant origin.